And at this point, I would like to introduce Deborah Niehoff, who is AFTD's Director of Research and Grants. Deb.
Good afternoon, everyone. They did. There. Now you can hear me. Good afternoon, everyone. Over the course of our 20-year history, AFTD has become a leader in advancing the science of FTD, hastening the day when this disease can be treated and ultimately cured. AFTD convenes leading minds in FTD science at events like our annual Holloway Summit and the FTD Research Roundtable. We've made strides in investigating potential biomarkers and treatments for FTD by partnering with organizations like Target ALS and the Alzheimer's Drug Discovery Foundation, and investing in initiatives like the Diagnostics Accelerator Project. Over the past 7 years, we've awarded nearly $25 million through our research grant programs to researchers doing innovative, leading-edge work, planting the seeds today for the breakthroughs of tomorrow. Today, well, thank you. Today, 3 of AFTD's recent grant recipients are on hand to talk about the work that they're doing and the promise that it holds for a better future. And I'm going to invite them to come up now. Dr. Matthew Nolan works at Massachusetts General Hospital, where he is a postdoctoral researcher, and he received a Holloway postdoctoral fellowship for his work in identifying and evaluating drugs based on a novel target that could treat TDP-43-associated FTD. Dr. Qinwen Mao is a professor at the University of Utah, and she received an award through the Diagnostics Accelerator Initiative, and she's using this to develop an assay that can be used to detect the FTD-associated protein TDP-43 in blood. And Dr. Emiliano Santarnecchi of Harvard Medical School and Massachusetts General Hospital has received funding from the Treat FTD Fund, made possible by generous multi-year commitments from the Samuel I. Newhouse Foundation and the Lauder Foundation, Leonard Lauder and Ronald S. Lauder, to develop a non-pharmacological treatment for FTD. And welcome to all of you. And Dr. Santarnecchi, I'd like to start with you, actually, and to ask you to describe this non-pharmacological treatment and what you are doing, what your study's about, and why it's important.
Absolutely. Second, if this works, I guess it works. You can hear me. First of all, thank you for inviting me. This is such a great honor. I was talking with our coordinator in Boston months ago. We were discussing how can we be more in touch with the community, talk with more patients, share what we are doing, and the same week I received an email from AFTD asking me to join this panel. So it was completely serendipitous, it was amazing. So I'm really glad to be here. The work we do in Boston is a bit outside the normal path of drug development. We also test drugs, but mostly we work on non-invasive neuromodulation. I think the best way to introduce that is to talk about invasive neuromodulation, which probably is more familiar for many people here. You might have heard of deep brain stimulation, which is a practice where we implant a device in the brain to treat certain diseases. If you find the right location and you find the right frequency of stimulation, you can really decrease symptoms almost immediately as soon as you turn on the device. These are very well used for Parkinson's disease, for instance. They're under investigation for depression, for Alzheimer's disease. But again, there is surgery involved, So over the years, over the past 20 years, we started developing non-invasive solution for neuromodulation. Basically what we do, we use a device that is applied over the head on the scalp, and it goes through either magnetic or electrical stimulation, but the results are kind of the same. We can, through neuroimaging data, so for instance, if we have the MRI of an individual, really be targeted and only target the activity in one specific region of the brain where we think the problem is or where the symptoms of the disease are coming from. So with this device, now we are trying to test a new potential therapy for FTD. My journey with this non-invasive neuromodulation started more than 15 years ago when I was using it for cognitive enhancement. I was trying to basically make smart people smarter, to put it in a very simple way. I was trying to boost memory, attention, logical reasoning, but then I started moving more towards the clinical field, and we started testing it in Alzheimer's disease in 2016 with a group at MIT in Boston. And it worked. The pilot data were amazing. Last year we showed that by doing specific stimulation at a specific frequency, 40 Hertz, with electrodes placed on the scalp, we were able to boost brain oscillatory activity, boost blood flow in the brain, so we increased perfusion, which, as you know, is also one of the characteristics of FTD. We have hypoperfusion and hypometabolism. And last year, in December, we published preliminary evidence of a decrease in tau protein measured with PET imaging after the therapy. So while I was developing that, I started looking around because there are a lot of things in common across neurodegenerative disorders. And FTD always fascinated me, and I found the same substrate that could resonate with the therapy that we were using. And so I was lucky to contact the foundation and through FTD and ADDF, and they were so nice to grant me money to run now what it is a multi-center international double-blind trial. So it's a proper phase 2 trial. We were trying to see if the therapy works in boosting perfusion, cognition, and hopefully even more than that. So I'm really glad to be here and be able to share more about what we do.
Thank you. And it's really excellent to have, in addition to the clinical trials of medications that you've heard about, to have a non-pharmacological treatment as well. And I'm just wondering if would this be a treatment that would be applicable to all types of FTD?
That's a great question. So in our trial, we focus on BV FTD, behavioral variant. It was a decision made together with the foundation to have a clear target at the beginning, but we already have pilot data with magnetic stimulation done in Europe in another group that I direct when we applied it to PPA. And the results were published last year in a small sample of patients. We saw very good results in combination with speech therapy. So hopefully we will push forward for that too. But in theory, it should work with many, if not all of the subtypes.
Thank you. And what is it like for the person who's receiving this treatment? What do they feel? What do they experience? Are there any side effects?
No, they love it. This is not me selling the therapy. That's usually the feedback we get, because you barely feel the stimulation on the scalp. It's really a tiny sensation, like tingling for 3-4 minutes, even though the treatment is for an hour sometime. But what they do, they come to the hospital and we watch movies together, documentaries with my team. Usually I sit there and I do it too, because movies are fun and we select the good ones. So you barely feel the stimulation on the scalp, and the feedback, it's really great. So usually patients ask if they can actually buy the device and do it at home, which is something we're investing on now, to build a headset that you can do at home instead of coming to the hospital. But yeah, no major side effects. For electrical stimulation, it's really maybe a migraine once a month, but it's mostly because the device comes with a cap to hold the electrodes in place, and so the pressure on the scalp for an hour sometimes gives you a light headache, but nothing more than that.
Thank you. Dr. Mao, let's move on to your study. And can you tell us a little bit about your project? And since your assay is an example of a biomarker, and I mentioned the development of biomarkers that AFTD has supported, maybe a good place to begin is with a definition of what a biomarker is.
A biomarker, basically, some protein particles in blood, CSF, that can indicate a disease or condition. It can be, in blood, CSF, or even digital, like CT, MRI, those imaging approach. But obviously, blood is a best option down the road. It's cheaper and most easily accessible.
Can you tell us a little bit about the biomarker assay that you're developing?
Right now, I have a project supported by AFTD on TP43 blood biomarker. I would like to also share with my career life experience. I have a long-term research interest in FTLD-TDP. I started that interest when I was a fellow around 2007, when TDP43 was first described. And then I joined Northwestern University, became an academic neuropathologist. I worked there for 10 years, examined a few hundred patient brains. Most of them, more than half of them, have FTLD-TDP. I was very fortunate to be able to work with a world-class neurologist. My mentor, my boss, is an internationally well-known neuropathologist with expertise in FTLD-TDP. While I working there, I quickly realized that there is a crucial issue in the field. The diagnosis only reviewed after the autopsy, after we examined the brains. Basically, when patients alive, they don't know what they have, what kind of proteinopathy is tau, beta amyloid, or TDP. So if you don't know what the proteinopathy is, a molecular mechanism underneath, you have no patient treatment. So I started focus on TDP biomarker development. The TP43, while I'm working it's a very complicated, interesting molecules. In disease condition, it can have different forms, different from normal protein. In different disease, they have different forms. For example, they form inclusions, deposits in FTLD-TDP, maybe like a potato, or in AD, and then maybe like a cauliflower, different from normal health condition. So how can we detect, grab, analyze? We can use antibody. Antibodies, our body, animal can produce some protein, can recognize those disease protein. So in the last few years, I screened 5,000 antibodies, and luckily, I called it pies from skies. I obtained 2 antibodies can recognize those disease protein, one in FTLD-TDP, another one in AD. They can detect much more pathology than the gold standard antibody available in the field. And even more interesting, with the support of AFTD, I was able to push the project move forward faster. And the AFTD support really gave me courage and inspiration for me to dedicate my focus to develop this biomarker. And right now, our team able to develop an immunoassay can be sensitively detect TP43 in blood from patient of FTLD, TDP, and AD. That's where we are.
Thank you. And maybe you might take a moment and tell us a little bit. We saw how drugs that are being developed go through phases before they're approved by the FDA. Does a biomarker go through different stages of development as well?
Yes, exactly. After we develop immunoassay in the lab, we first need to do the verification in the lab, and then validation in the pathology lab, followed by FDA approval, and then we can launch. I 2 years ago, I moved to University of Utah because the pathology lab, like ARUP, is the second largest nonprofit referral lab of the country. So it has a platform to launch the test. It has a platform connect to most of the clinic hospital of the country. So it can be best useful to make it faster to the commercialization.
Thank you for that explanation. Dr. Nolan, you are a postdoctoral fellow, so you're at the very beginning of your research career. And I wondered if you could share with us what your motivation was for getting involved in FTD research.
Yeah, absolutely. So firstly, I'd just like to say thank you very much to AFTD for inviting us. It's a real honor to be able to do things like this. And I'm personally really grateful for the continued support that AFTD provides our work. So what got me into it originally was a master's project that I started in, I think, 2012. And that was actually on a different disease. And then over time, I became gradually more and more interested in the concept of RNA binding proteins and what they do and their significance in neurodegeneration. And the thing with RNA binding proteins is that under normal circumstances, they should be localized or within the nucleus, which is where your DNA is stored. But for reasons that we don't really understand in the disease, they move from the nucleus into the cytoplasm. So what that does is it confers a problem on two fronts. Firstly, you have the presence of these proteins in a place where they shouldn't be, which confers toxicity. Secondly, you lose the normal function of those proteins from the nucleus, and that has a knock-on effect on the expression of thousands of other genes. So my work now is really based upon identifying ways that we can modulate the expression of another gene, another protein called stathmin-2, and which we believe is very important in the TDP-43 mediated disease process. And so what I've done is I've screened thousands of drugs for their effect in being able to increase the level of this stathmin-2 protein. And the advantage of an approach like that is that many of these drugs are already FDA approved, and that makes it easier when it comes to the approvals process because we know about their dosage and their safety profiles and their drug-drug interactions. And so that is really what our lab is focused on, is understanding the significance of loss of RNA binding protein function from the nucleus.
Thank you. And can you tell us a little bit more about stathmin-2? What does it do in a healthy neuron?
That's actually a really good question. So stathmin-2 in a normal healthy neuron is responsible partly for that neuron's growth, but also repair following injury. So we think that when you lose stathmin-2 from the cell, that it inhibits the ability of the neuron to recover from some kind of insult, whether that's an acute insult, i.e. you physically injure the neuron, or whether it's a degenerative insult, as would occur in FTD or ALS.
And you mentioned that there are other genes that are affected by TDP-43 and when it gets mislocalized. And are any of those promising targets for drug and biomarker development as well?
Yes, we believe so. We're really at the beginning of our understanding of this. The progress that we've made in the last just 4 or 5 years has been really quite significant. And we're really just now starting to understand what is the consequence of loss of TDP function from the nucleus. But the reality is that there are actually thousands of genes that are affected when you lose TDP-43 from the nucleus. And understanding which ones of those genes are really important and to what extent they are important, that is the focus of a very large degree of research around the world. But for stathmin-2 in particular, there's a lot of excitement now about stathmin-2. Everybody wants to talk about it when we go to scientific conferences. It's a privilege really to be able to work on something that is really, I feel, at the forefront of where we are at with FTD research now.
Thank you. I have have another couple of questions for all of you, but before we get to that, I want to open it up to any of you in the audience who would like to ask a question. And if you are part of the virtual audience, if you want to put that in the AFTD app, those will appear up here on stage for me, and I can ask those as well. And we already have a question. Go ahead.
For the young lady in the middle there, have you ever looked at troponin for being a biomarker? And the reason I'm asking, my mother died of at that time they called it Pick's disease. They called it Pick's disease, but it was FTD. And her troponin levels were always high. I have been to the hospital on numerous occasions for this, that, and other, and every time they want to put me in the cath lab because my troponin level is high. And I have talked to other people with FTD, and they had the same problem.
Oh, that's a very important information. Actually, my goal is develop like a multiplex. I will put that, I will look into it and put that on the list.
Mm-hmm. Thank you. Thank you for that observation. Is there anyone else who would like to ask a question?
This is for Dr. Nolan. Mm-hmm.
Matthew, it was fine.
When you mentioned physical damage of a neuron, structural damage because of, are you talking, how would that happen? Would it be through a head injury, traumatic brain injury? What would the chemical injury, how would that happen?
Yeah, so we can, in the lab, we can simulate those kinds of things quite easily via like physically cutting a neuron. In patients, my honest answer is I'm not actually completely sure. But I mean, if you, the thing is you have many types of neurons, and they probably respond differently to different kinds of insults. In fact, they do respond differently to different kinds of insults. But what we know is that even in the kind of work that we're doing at the moment, I believe it has implications not just in FTD and ALS and degenerative conditions, but also things like kind of spinal cord injury and, you know, physical damage to the neurons.
Mm-hmm. And Dr. Nolan mentioned a very important point, and that is another really hot area of research right now is what triggers this entire neurodegenerative process. And it may not be the same thing for every single person, but it is a really interesting area of research right now. Other questions?
This one might be really for FTD, AFTD as much as the panelists. Repurposing drugs that have already been approved is great. The challenge, though, is who's going to pay for the clinical trial and who's going to run it, and etc. And I just wondered if there's any, maybe it's a bit early yet, but if there's any thought about how a molecule, if you find it, can be developed.
That is a fantastic question. And I will point out that our Treat FTD Fund funds clinical trials of both novel and repurposed drugs. And in fact, one of the other funded projects that's in play and active right now is a repurposed drug. So we can kind of get the ball rolling, but you're right, that is a challenge then to take it a little bit further. But you have so much of the early work done because all of the toxicology and all of the information about how the drug's absorbed and processed, that's already been done. Any of the panelists want to offer anything else on that?
I've been involved with the similar kind of repurposing drug adventure with ADDF. We're trying to repurpose rotigotine for Alzheimer's disease with a colleague in Italy. Works beautifully, strong preliminary data. To me, when you enter that space, it's also a big conversation about IP. Who owns what, which is something I hate about my job, but you have to have many of those conversations. Sometimes it slows the process down a lot. So I think we need to put equal thoughts into where we find the money and where we remove the block at the beginning of the conversation, because a lot of those conversations about rotigandin, I'm pretty sure has been going on for a decade before we connected the dots. Many people gave up before us because they were like, how am I going to pay for the thing? They're not going to let me, Pharma X, use that thing. We need to work on that, I think. That's my opinion.
And I think we can take one more question. Anyone else? I think we have another question up here. Oh, okay, go ahead. I'm sorry.
Yes, I was told that once the brain dies, there's nothing you can do. Is anybody doing any type of research? Is that true? Is there any way to bring the brain back? Or is everything just on trying to stop or slow the progression of FTD?
Yeah, I think that's. I think there are lots of people who are actually trying to induce what we call neurogenesis, so the birth of new neurons. It's very, very difficult, but that is definitely something that a lot of people are doing, because there's a big difference, of course, between preventing the progression of a disease and reversing it. So you make an excellent point, but there are lots of people that are working on similar things.
Anyone else on the panel?
I think one way to look at it is that, as Dr. Nolan was saying, it's true that so far we see neurogenesis only in a few parts of the brain, very small regions in the brain, but that's not the only solution, right? To create new cells. You can rewire around the damage. You see that in stroke. When you have a stroke at the beginning, the brain is not functioning properly. You go through rehabilitation. If you make a prediction the very few days after a stroke, you would say this patient is not going to do anything for the next 10 years. But then you go through rehabilitation and you start moving your hands again, legs, you start talking. That's the brain rewiring through plasticity. So we have ways to boost plasticity in the brain. So basically you use the same neurons, you just make them talk to each other differently and find shortcuts to do the same job, but in a different way. That we can work on, and there is hope there, if that helps.
This is really exciting. I want to give all of our panelists a chance to wrap up, so we won't be able to take any more questions here, but they will be present at the break and at the reception. So if you didn't get to ask your question, please go up and talk to them. Please send AFTD your questions, and we'll do our best to answer them. But I want to have a chance to ask you one final question, each of you, and that is, what is it right now that you're most excited about or most optimistic about in FTD research? And Dr. Nolan, why don't we start with you?
So I think the thing that I'm most excited about is what Dr. Miller mentioned earlier in this morning briefly, which is antisense oligonucleotide technology. And these are short lengths of DNA or RNA that we can chemically modify to either repress or induce the expression of a gene. And you might have seen just last week that the FDA approved tofersen, which is an ASO targeted against SOD1 ALS. And I really think it's only a matter of time before these kinds of things start making it into the FTD space. And broadly, in terms of genetic medicine, I mean, there are things entering the clinic now and in late-stage clinical trials that even just kind of 10 years ago would have been considered total science fiction. And these things are really happening now. And it gives me a level of optimism that before I wouldn't have had. But these things are happening, and they're happening very fast. And I think we have every reason to be very optimistic about the future of FTD therapeutics.
Excellent. Dr. Mao, what are you excited about in biomarker research?
I have 2 points. One, it will be able to make diagnosis on living patient, hopefully in the next few years. And another one, those antibody can recognize those disease protein, so it can be a monoclonal antibody medicine, just as those recently approved by FDA, those monoclonal antibody against AD. TDP-43 antibody also have this similar potential. And I want to add a little bit my personal point to Terry's question, whether we can reverse the disease. You know, I have some experience using mouse model when we when the mouse has pretty bad pathology. After remove the deposits, actually the animal behave much better. They can walk faster on the wheels. So hopefully that can happen in human being, but need more experiment.
And Dr. Santarnecchi, what are you excited about?
Yeah, I hinted to this a bit before when I was talking about porting, like moving the technology from the hospital to home and developing this new portable headset for brain stimulation. What we are working on now is a platform that comes with this device that not only allow you to do brain stimulation at home, but also record brain activity anytime you want in a painless, non-invasive way. It's the idea to build a platform around each patient because we base a lot of our assumption clinically, but also at a scientific level on little data. We think we have a lot of data, but you are lucky if you see your neurologist every 6 months, maybe once a year. You get an MRI scan once a year. Rarely you get brain waves recording or other type of data. I'm trying to create a platform where you can get this data at home anytime you want. So we focus on each patient because each patient is different. And also we would like to implement, as the presenter said this morning in the keynote speech, you are the best clinicians. So we want to hear from you. We want to generate a platform where both the patient and caregiver can send their notes every day so that we absorb the data and maybe we will capture subtle differences that over the years will generate tremendous knowledge to personalize treatments and hopefully make even further strides in the search for a therapy.
Well, thank you. Thanks to all of you. We really appreciate you coming and telling us about your research today, and we want to thank you as well for all the work that you continue to do furthering FTD science, contributing to hope, and advancing the path to approved therapies. And at this point, I'd like to welcome Matt Sharp back to the stage.